All right, guys, let's go back to our lecture. Um, yes. Okay, so we talk about cell, we talk about cell membrane, and then we talk about membrane protein, and from there we talk about protein synthesis. And from there we know that all cells come from the same cell. All cells have the same set of protein, same set of the chromosome, but they express different protein. Why do they have different structure? Because they have different skeleton. Why do they have different skeleton? Because those skeletons are protein. Intracellular protein form this cytoskeleton. So these proteins provide the structure, provide the function, provide the communication between cells. So that is what makes a cell unique. Otherwise, every cell will be the same, right? They are so different, just like human, right? In our society, different person produce different function. You have your calling, you produce different products, cell produce different proteins. So that's that. I want to use this one to lead into a very important concept that is the membrane protein. So here is one of those membrane proteins. So you have different kinds of proteins. Some of those right here, some of those released outside of cell, some of those attached to the membrane. And one of those membrane protein is called sodium potassium ATPase. This is a protein. What does this protein do? First, anytime you learn about a new term, you ask location and the function. Where is it? What does it do? What is this sodium potassium ATPase? Where is it? It's on the membrane. It's on the membrane. Go back to the very first membrane protein we talked about. Right here. Yeah. So here is a membrane. This is a membrane protein. This is the one that we are going to talk about. One of those use ATP, and so that is this one. This is the most important one. Even though cells may produce different protein, but every cell has this one. So this is universal. Every cell has it. These proteins anchor on the membrane. Where is it? It's on the cell membrane. It's a membrane protein. What does it do? Location and function. What does this do? This one will use ATP as a fuel to pump. Every time when you have ATP, as long as it's charged, it uses this ATP, use its chemical energy, ATP become ADP, triphosphate to diphosphate, release a phosphate, use that chemical energy to pump. What does it pump? It pump 3 sodium, those are sodiums, outside of the cell, and it will pump 2 potassium into the cell. So that's what it does. So location is on the cell membrane, it's on the plasma membrane. And what does it do? It pump 3 sodiums out, pump 2 potassiums in. Is that really matter to remember those numbers? Yes. Is that really that matter to remember which ions they pump? Yes. Is that really matter to know the direction that they pump to? Yes. So you got to know that with this one, because this is universal, every cell has it. And so every cell will have the function provided by this protein. So you got to know that what does this pump do? 3 sodiums out, 2 potassium. You're gonna see this in the quiz. What does this sodium potassium pump do? It pumps 3 sodiums out, 2 potassium in. With this pump, there's 2 consequences. One consequence is that because it pumps so potassium into the cell, sodium out of cell, it will make this intracellular environment. to have high concentration of potassium because you pump potassium in. When it pumps out, basically free floating around, this extracellular is just like ocean, right? It's shared by every other cell. So it pump out everything out there. Basically, it's free diffuse everywhere. But when you pump things into a cell, you capture them in the cell. So you have high concentration of potassium inside of cell. So that's the first consequence. It causes high potassium concentration intracellularly. If we look at this one tell you about the ion, potassium, sodium, calcium, chloride, ion concentration, different concentration intracellularly versus extracellularly. You can find different number right here, but in the end, if we summarize it, basically just one thing. The only ion which has high concentration intracellularly, that is potassium. Everything else are high concentration extracellularly. So with this pump, it causes high potassium concentration intracellularly. So that's first consequence. Which ion has high concentration inside of cell? A quiz question. Which one? Potassium. Okay. And do you find this in red blood cell? Yes. Do you find this in skin cell? Yes. Do you find this in neuronal cells? Yes. You find this in every cell. Every cell has this pump. Every cell has this protein. Every cell has high concentration of potassium intracellularly. So that's one consequence. The next consequence is because that the pump charged, positive charged ion, but different quantity. You pump 3 out, pump 2 in. So these numbers are not the same. And so you pump more positive charge ion to the outside. You pump less positive charge ion to the inside. And so in the end, you will have more positive outside, less positive inside. You create the disparity. You create that intracellular is more negative compared to the ocean, the outside. Outside, everybody share it. So this ion free diffuse everywhere. But inside, you don't really pump that much inside, right? You pump only 2. So you make the inside is more negative compared to the outside. So that's the second consequence. So here I summarize these two consequences. You will make the membrane potential, the diversity, the disparity, make it more negative inside compared to outside. You also make intracellular concentration potassium to be high. So that's that. All right. So that is the foundation of electrophysiology. It's all due to this pump. So that is a consequence. When we measure the membrane potential, this potential refers to the voltage. We measure it when we talk about the membrane potential. We talk about the membrane potential inside in this side of the membrane. Why is that? Because outside are shared by everybody, right? But inside is unique. The membrane potential of this cell we measure is inside, and we will always get a negative number, negative membrane potential. So that is the membrane potential, and every cell has it. So every cell has a negative membrane potential. All cell has a negative membrane potential. So that's that. All right. Not only the so not only that membrane has a pump, membrane also has a membrane protein. So several of these membrane protein will form a channel. So here showing you how do we use this membrane protein to form a channel. This is the protein. We fold it, we form a sheet. This sheet fold it over, anchor on the membrane. We form a channel. With this channel, this is very much like a straw. You have the straw to drink for example, your Starbucks, right? Or your juice. This is membrane, this is in your cup, this plastic. Water cannot diffuse across. Water can, but in your cup, water cannot. And you need to, if you want to drink something, you got to answer a straw, and that is that protein. In our membrane channel, this channel, it will select which can go through, which cannot. These channels they are different kind of channel. They are different kind of ion channel. And some of those are gated, meaning that its opening depends on a condition. It's gated, it's closed. And you can open it by a key or by knocking a door or by doing something, right? So it's gated. And there are some other proteins, there are some other channels that's not gated. When it's not gated, we call it a leakage channel. Our body has cells has multiple like leakage channel. It allows things to go through. Yeah, so that's that. So we can have gated, we can have non-gated. Not every cell has the same set of ion channel. So this will be different depending on the cell. Every cell has this though. Every cell has this sodium, potassium, ATPase. So this is universal. But if you're going into some other different kind of ion channels, it will be dependent. Different cell has different ion channel. And they also have different kind of ion channel, either non-gated, non-gated ion channel, or gated. When we talk about the gated ion channel, we can have ligand gated, like a chemical binds on it to opens it. We have ligand gated ion channel. We also have voltage gated ion channel because these ion channels are located in the membrane. Membrane has a membrane potential. So they may be sensitive. Some of those may be sensitive to the membrane potential. So if the membrane potential is disturbed, it may cause it to open or it may cause it to close. So it may be membrane, voltage gated. We also have mechanically gated. So this one probably should put it under the gated channel, mechanically gated channel. For example, what are the mechanically gated? Mechanically gated meaning that its closing opening depends on mechanical change. And some of those are sensory receptor, sensory neuron. For example, that your finger, right? When you touch the table, you feel it. Why is it? Because you provide a mechanical deformation to your membrane channel. So when you touch it, you distort it. And by distort it, you cause it to open. Ion will flow through, cause stimulation. So a lot of these skin sensory receptor are mechanically gated ion channel. So they are different kind of ion channel. Yeah. And they are all protein. Why are they different? Because cell produce different protein anchor on the membrane to do their spatial activity, right? Spatial function to perform its differentiated function. Yeah, so that is the protein on the membrane. Among these different membrane channel, membrane protein, different ion channel, there are some ion channel are so unique that make the cell to be so-called excitable cell. So that's what we are going to talk about here. Oh, before we talk about that, let's just have a picture showing you these different ion channel. So you can have a always open, like a liquid channel is always open. Sodium is selective. So this one is for potassium. This one is for sodium. Not every cell has it, but some cell has it. You can have a ligand gated. So for example, a ligand can be a neurotransmitter. Maybe we will talk about that. Yeah, a chemical basically binds on it, and it will cause it to open. This one is for sodium, so it's a ligand-gated sodium channel. We can have a mechanical pressure, like the receptor on your skin, right? With the pressure open, touching it, it opens it, sensitive to the mechanical force. It will open, allow sodium to, or like a chemical to go through the ion channel, and or voltage. So it depends on the memory potential voltage. Yeah, so we have different kind of ion channel. And some of those are so unique that make the cell to be an excitable cells. I think that is, yes, that is this one here. So not every cell is excitable, but every cell has sodium, potassium, ATPase, right? Every cell has this memory potential. Every cell has it, but not every cell is excitable. What makes the cells to be excitable? Later, we will talk about what does it mean by excitable. Excitable means that it will generate an action potential. So, what makes a cell to be excitable is that these cells produce a unique protein anchored on the membrane. These unique proteins are sodium channels, are voltage gated here. Not every cell is excitable, but those excitable cells got to produce those proteins, produce two type of protein, a voltage gated sodium channel and a voltage gated potassium channel. So got to have that. Not every cell has these two though. Only unique cell. What are those unique cells? Muscle cell has it. Neuronal cell has it. They have these two channels. When they have these two channels, they are excitable because these channels are voltage gated. They are sensitive to the membrane voltage. And when they are sensitive to the membrane voltage, they can get triggered by stimulus. And when they have that channels, when they produce these proteins anchored on the membrane, they will do one thing very unique. That's called an action potential. They will generate an action potential. So not every cell has it. Only muscle and the neuronal cells has it. Why do they have it? Because they synthesize these proteins. So let's look at these proteins and to see how do they form an action potential. So what is an action potential? An action potential is a membrane potential. Is a membrane potential. So membrane potential usually is negative, very stable. Is negative inside right here. A membrane potential is negative inside. It's always like that. It's always not change. But action potential is a quick change. Okay. So for example, let's look at the action potential here. So action potential is a quick change. So you can see that the membrane potential is always negative. In this case, it's a negative 70 millivolt. So it's negative all the time, but it can generate a quick change. So it's an action potential, location and function. Where is it? On the membrane, on cell membrane, on the plasma membrane. What does it do? It makes the membrane potential to be quickly change. And that membrane potential can travel along the cell. And so that's very important to conduct an electric signal travel along the cell membrane. For example, that we know that these membrane are continuous covering the whole cell. For example, this is a neuronal cell. You then generate that action potential, membrane potential change. It can travel to the end. The cell can be very long. Your skin has a neuron in your spinal cord. So you can see that fingertip has a receptor. So you can see that the neuron, the cell length can extend from your fingertip all the way to your spinal cord, right? So that's very long. And so action potential, and it also travel very quick. So it can travel from the fingertip all the way to your spinal cord. And so that is the memory potential, that is the action potential. That's how we transduct the signal. So why is this action potential? This action potential is achieved by those two proteins, voltage-gated sodium channel and the voltage-gated potassium channel. So the action potential has several components. If it's not activated, it will be the resting. You will have to have some stimulus. You got to touch it, stimulate it, electric shock on it, do something on it, right? You have to stimulate it to generate an action potential. Your stimulation doesn't have to be very strong. As long as that stimulation is above the threshold, it will trigger it. So you have stimulation above to change the memory potential to be above the threshold, and then it will trigger a action potential. The action potential has two phases. One is called depolarization. This green phase is a depolarization phase. This one, depolarization. And then you have repolarization. Why do we call it depolarization? Why do we call it D? It's because that the memory potential is polarized, right? The memory potential is negative. And what this one does is to reduce that polarization, to make it less polarized. And so we call it a depolarization. After a depolarization, you will have a repolarization to make it a polarized again. It's very short, couple milliseconds, and so that's an action potential. These are achieved by those two proteins. So let's look at how does we do it. So here is the some stimulus. You have to stimulate it. If the stimulus is not high enough, your hearing is part of that nervous system. And so when I talk, I have to be loud enough, right? But it doesn't have to be super loud, but you have to be loud enough to stimulate it, right? So when you touch, yeah, you gotta be strong enough, right? It doesn't have to be super, super strong. You are sensitive, so you can feel it, but you have to stimulate it to be above the threshold. So that's the threshold. So you have to, if you only stimulate this, it doesn't really cause damage, cause stimulation, cause action potential. If you stimulate like this one, it doesn't really, it's not strong enough. But you stimulate it above threshold, you trigger it. So that's action potential. That's the threshold. Now, after you provide that stimulus above the threshold, you trigger an action potential. It has two phases: depolarization. Sometimes we have an overshooting, and then repolarization. So that is achieved by two proteins. The first one is voltage-gated sodium channel. So when it's above threshold, these voltage-gated sodium channel open. And so let me draw it. Let me draw it. Pencil tool. All right. So say this is the cell, right? This is cell. And you have a voltage-gated sodium channel right here, right? And so that's a voltage-gated sodium channel. Now, when it's open, it will make the sodium freely diffuse. So this is sodium freely diffuse. It's ion channel, so it's open, right? It's above threshold. Now it's open. Sodium can freely diffuse across. It's free now. Sodium can free diffuse. And this net effect of that diffusion is driven by concentration gradient. So the net effects of that sodium movement is from high concentration to low concentration. All right. So my question to you is that when this ion channel open, will most of sodium move out or move in? Will sodium move out or move? They'll move across, right? Free. But the net effects is from high concentration to low concentration. Where do you see high concentration of sodium? Keep in mind that the only thing high inside of cell is potassium. So where do you have high concentration of the sodium? Outside. So when this channel open, where does sodium go to? Sodium will move from high concentration to low concentration. Sodium will move in. So that's what's happening. When you have a stimulated, you open a voltage gated sodium channel, sodium comes in very quick, a lot of sodium, because that's from high concentration to low concentration. They just diffuse in. And the sodiums are positively charged ion, so they will bring in positive charged ion. Inside was originally very negative. Now you bring in some positive ions to make it positive, Sometimes it's overshooting, but make it positive. So you provide that depolarization. Yeah. So that is a depolarization. And the quiz question can be that which ion channel contributes to the depolarization phase of an action potential? That will be the voltage gated sodium channel. So when this channel is voltage sensitive, so it will open when you stimulate the membrane to be above the threshold, but it will close when it reaches to the neutral, when there is no such difference. Their positivity is trying to eliminate polarization. They do it. They complete the journey. They complete the job. They will close. When they reach to a neutral membrane potential around zero, they close up. Sometimes they're overshooting, but they close up. So this close. When it's closed, another ion channel that I mentioned earlier, that is a voltage gated. That's right here. That's joy here. So that's a voltage gated potassium channel. So this one allow potassium to free diffuse across. This one, after this one close, when the membrane potential reach to the neutral, this one open, potassium channel open. Now the question is, when this channel open, would potassium move in or move out? They will free diffuse across, but their net effects is from high concentration to low concentration. Where do you see high concentration of the potassium? Inside of cell. So it will diffuse out. This is positive charge ion. So they diffuse out will make it inside to be less positive. So they will regain that polarization. So the next phase is the repolarization. Is achieved by voltage gated potassium channel. When it open, it allow potassium ion to move out. When they move out, it will make intracellular to be negative, like here. So it will move out, it will make inside to be less positive. It will be negative, right? So this is a repolarization. Why do we call it a repolarization? Because you make it polarize again, like before. So that's a repolarization. So this is the action potential. Very simple. Is phenomena occur on the membrane. Not every cell produce action potential. Those which can produce action potential, we call them excitable cells. Why do they become excitable cells? Because they produce two other proteins. not every cell has these two proteins. What are these two other proteins? A voltage-gated sodium channel and a voltage-gated potassium channel. And with these two, they will make them excitable. We typically see these excitable cells in the muscle cells and in the neuronal cells, and they will generate action potential. So that summarizes it. Let me know if you have any questions. And with this one, we finish the cell portion, and then we will move on to another topic. Okay.